Hello, everybody. My name is Leslie Espinosa Campomanes, and I have been working in the Luo Lab alongside David Wong for this 2021 BISER program. It is a pleasure for me to introduce to you all with my research, which I called Yummy, Feeding in Development, How HRP Neuronal Circuits Influence Mice Behavior at Different Ages. Before I start talking about my project, I want to discuss a little bit about homeostasis, which is the self-regulating processes that any living organisms adapts to maintain internal stability while adjusting to conditions for optimal survival. One very common example for that is how our internal body temperature changes. Another crucial one is how we can balance food supplies, energy supplies, which is met by feeding. One of the abilities we inherited since we were born, as you can see in this image. But feeding and hunger, these sensations are felt by the AgRP neurons, which are located into the arcuate nucleus of the hypothalamus, as you can see here. These are the AgRP neurons. And although we might think that as hunger and feeding is something we are innate, the AgRP might be working since the very beginning of our lives, right? But actually that's not the case. And this research has shown that. According to this graph, this was a neonatal ablation of AgRP neurons, right? So the red dots and red lines are the ones, the mice with AgRP neurons ablated neonatally. And as you can see here, their licking behavior hasn't changed drastically. They are basically very similar than the wild type mice. On the other side, when you ablate AgRP neurons as adults, the red dot and lines are very different from the wild type mice, right? So their licking behavior changes drastically, suggesting that the AgRP neurons are necessary for hunger in adults, but you can ablate them in neonates and seemingly there won't be any drastic effect. So with this motivation, in our project, we are trying to discover when the AgRP circuits target different regions of the hypothalamus and when this matures. Then, what behaviors are driven as a result and how those are relevant for the mice's ages. So in order to do so, we have two approaches. First, we're tracking the AgRP axonal projections. And to do so, we have a series of protocols, starting for perfusion, which is when we get rid of all the blood present on the tissues so that we can examine the brain. After that, we do a collection of brain sectioning, and as you can see in this figure, and then we do antibody staining to these brain sections with a primary antibody with NPY and TD tomato tracking AgRP cells. With those staining and the secondary fluorescent antibody, we go to the imaging processing, and then the images can be labeled green for NPY, red for TD tomato, and blue, as you can see, is DAPI, which is the cell's nuclei. Finally, with all of this data collected, we can send it to the Elastic, which is an artificial intelligence software that basically help us segment certain areas. In our case, and for our purposes, the axonal projections, and compare it with the background of the image. The other approach we are looking at is the neonatal behavior. And this is because we want to understand what types of behaviors the neonates have. So to do so, we have developed a 24-hour recording setup with this cage setup, which is called the K for me. And we do it during daylight, sunset, sunrise, and during the night. This setup has shown that, well, the pups interactions are mainly resting and moving in the nest and suckling, but they might not be outside of the nest, as it is just 1.4%. In contrast, the mom's behavior is also staying with the pups most of the time, outside the pups to get some water and food, 12%, and going to the surroundings, which is something that they just reflect at the beginning of the change of the setup of the cage. So exploring what's going on. All of this is because we really want to know whether a certain pattern behavior is implying that the brain is telling the neonates or the moms to do so, and how these behaviors develop in time and according to maintain homeostasis in their cages, right? So yeah, this is the behavioral side, and we keep working on this using DeepLabCat, which is a software for animal pose estimation, can better and accurately help us telling what behavior is what behavior and how often that's happen. As you can see, there is a lot of intersectionalities of the biological sciences as well as the environmental effect on the animals. And for me, at least, this has been such a wonderful experience to have to work in both sides simultaneously. So thank you so much for your attention. I hope you enjoyed this presentation, and I am looking forward watching yours too. Bye.